Let's see how many people do we have. It's rising, rising.
Mhm.
Uh,
What is IPOH in the background?
that's my hometown in Malaysia. I could change. Let me see. Hey, you recognize this, Bob?
Oh, nice.
Right?
Class, oh yeah, no.
Yeah, it's a resistant base.
An
Is that
X-wing.
Yavin? Is that the moon? Yeah.
Yeah. Where's Chewbacca, Bob?
Chewbacca? Well,
Yeah. Where's your best friend?
where did he go? Right there.
Hey.
Shall I leave that on? No.
Why not? No?
We've got 159 participants signed up.
Yep.
Wow.
Uh-huh.
Yeah. Okay. So 10 seconds, 8. Okay. Why don't you start broadcasting if you want, Con? All right.
Yeah. It's already on broadcast now. Okay, cool. Maybe like before doing this, like, thank you everyone for joining us today for our first solid state NMR and DNP like Zoom in session. And I'm Kong, a postdoc in Bob's Griffin group at MIT, and I will be doing like some IT support and doing housekeeping and responsible for the Q&A session later. Before doing all of this, I would just like to share with you the PowerPoint slides and PDF and keynotes that was actually provided by Xander, our first speaker today. And you can, you already like got the mail that I sent via the mailing list. If you have not received the email, in the mailing list, like please subscribe to it, because like there's an important note to that. Like after you subscribe it, once you put in your email, they will send a verification email of which you have to click on it to confirm it. You know, if you don't find a verification email, go to your inbox or junk mail, it could be there, because like that will be our main way or main channel to distribute like the emails or announcement in future. Okay, that will be it. And then so our speaker will be speaking for 40 minutes or 45, and like followed by Q&A sessions. And we are trying to do, we try to adopt like both ways of asking questions. You can either stay anonymous and use the Q&A function at the bottom of your Zoom chat room. Can you see the Q&A? So like you can either identify yourself, or you can click the anonymous function and like type any question you like, and you can actually like ask any questions like anytime during the presentation or after presentations. And one of the nice function that we can use via this webinar function is that after you type the questions, and if other participants that would like to ask the same questions, then can actually put a like or vote on the question. And then those with the most likes and votes will actually get asked first. That's like if you want to type. And on the other hand, for those who want to speak, you have to raise your hand. I think like most of you are very, very familiar with the raise hand feature. So raise your hand, and you cannot unmute yourself, but if I see you raise your hand, I can unmute you and then you can speak. Right? Okay. So and the other thing is that, so if you have anything that you want to communicate with the panelists or to all the attendees, you can do it via the chat, via the Zoom, the chat room, and you can like just type. And if you have anything, you can private message, or if I'm doing anything wrong that or suggestion, whatever, you can just like type in your message there. Recording wise, so like we will only record our session if we have the permission from our speaker. And our first speaker, Xander, is very generous, very kind. He says that do whatever you want, record it, put it on the group website, or put it on the website or whatever. So like we are recording the session now. So if you are concerned about privacy, you don't want to identify yourself, so don't speak or whatever. But we will, after the session, I will put everything on the group website. And of course, I will send out all the information via the mailing list. All right, so I will then pass on to Bob, and then Bob will do the introduction.
Okay, thank you very much, Con. The purpose of this little Zoominar webinar is to replace the fact that so many of the meetings that we have all attended for where we communicate with one another, we see one another, we exchange ideas and so forth, have been canceled. For example, the ENC this year, which was in Baltimore a few weeks ago, was rather sparsely attended. There were only about 500 attendees. Normally you get about twice that number. Uramar has been postponed. The Hype 20 DNP meeting, hyperpolarization meeting has also been postponed. And several Gordon conferences and keystone meetings have also been postponed. So Kong and I were talking the other day, about a week or so ago, and his brainchild was to start a webinar series, which you're starting to look at right now. And the idea is to partially replace these scientific conferences and personal interactions that are so important, so we exchange ideas in science. So I hope I don't need to really explain this. Everybody really appreciates this problem. Now this webinar is really an experimental endeavor. It was created in less than a week, about 99% by Kong, with a little help from me and also from Michael. And we're going to start it initially with a bunch of established investigators who will do the presentation. So this week, Xander Barnes from the ETH in Zurich is going to do one. And next week, we will have Matthias Ernst, who's also from the ETH, and Songyi Han from the University of California at Santa Barbara. And what we hope to do in the very near future, quickly, is to transition the presentations to include postdocs and students. So if you're a principal investigator listening to this meeting, this conversation, and I see there are about 240 people participating at the moment, please recommend to us a postdoc or student whom you think would be eligible to do a presentation. So the idea is we're going to have talks that are going to last approximately 1 to 1.5 hours on this seminar. And this would allow time for maybe 2 to 3 talks for approximately 30 minutes, including questions. And we hope there will be a lot of questions. Don't be shy. If you expose your ignorance, you really will learn something. Kong has already discussed recording. Please do not do this unless you have the permission of the speaker. So, okay, that's, I think, all of the background information that I needed to share with you, and Kong has also covered several points. And the speaker today is Alexander Barnes, and otherwise known as Xander, and he is a graduate of MIT. He received his PhD in our group. He built a couple of 250 gigahertz gyrotrons. He did a lot of really interesting experiments developing probes, low temperature technology, as well as many interesting applications. After he finished his PhD, he moved to Stanford for a couple of years or so, where he was a postdoc with Annette Cegelski, working on amyloid systems and several other things. And then he became a professor at WashU in St. Louis and achieved tenure a year or so ago. But about that same time, the ETH in Zurich started looking for a replacement for Beat Meyer, who was retiring, and Xander applied for and received the offer, and I think he started there in September of 2019. And today he's going to tell us about these very interesting spinning spheres that he's been working on, replacement for a cylindrical rotor, as well as chirped microwave pulses for DNP and in-cell NMR. And what he's trying to do, he tells me, is to move DNP to 1.2 gigahertz, what's 790 gigahertz for the electrons, and he wants to spin at greater than 200 kilohertz. And what we'll hear today is about the initial steps to achieve these goals. Okay, so Xander, the floor is yours.
So thank you very much for the kind introduction, Bob, and thank you very much, Kong, for the idea of putting this together and the kind invitation. I'm really excited to share with everybody the progress that my group has made over the last 5 or 6 years. So what you see on this title slide is a 28 Tesla magnet manufactured by Bruker that we're expecting to be delivered to the ETH. And our goal is to build a gyrotron that will allow us to generate pulsed and chirped microwave pulses at 792 gigahertz for pulse DNP transfers. Our sample containers are these spherical rotors. Inside the spheres, we're putting intact human cells, and our structural target of interest is a membrane protein, specifically protein kinase C. And right off the bat, here's the challenge that we see ahead of ourselves is sensitivity. All right, so if you want to do an in-cell NMR experiment, The main challenge is that the systems that you're looking at are much more dilute than you would have in a purified sample using a reductionist approach to structural biology. We will be using high magnetic fields, dynamic nuclear polarization, and in some cases, access to very low temperatures below 5 Kelvin to increase that sensitivity. So we want to determine these structures when the proteins and the drugs are within the actual intact human cells. We want to make very precise distance measurements of the ligands, in this case, that are activating protein kinase C. Here I show you bryostatin. And we want to make really precise distances between spins on bryostatin, let's say between fluorine or carbon, carbon, nitrogen. Because if you make a precise enough distance, you will see, as we have seen already, that of course the drugs and the proteins don't just occupy one single conformation, they're plastic. There's a lot of conformational heterogeneity, and this is really one of the secrets of biology. We want to get at that conformational heterogeneity in the conformational ensemble by making very precise distance measurements that can actually reflect and determine what that conformational ensemble is. And even more demanding in terms of sensitivity is that we don't want to only measure distances, we'd actually really like to characterize what the tensors are around all of these spins of interest. Here's a nice image from Leonard Mueller's tensor view. And this requires even more sensitivity. So, you know, NMR spectroscopists are very hungry for sensitivity. The more that we can deliver to the field, the higher impact we believe our research will have. So I think there are a few constants in the field of NMR, solid state NMR, at least the constants in terms of the first derivative. So we always want to go to higher magnetic fields. Higher magnetic fields give us more resolution and more sensitivity. We always want more sensitivity any way we can get it. Of course, dynamic nuclear polarization is one of the most effective strategies right now. And of course, we also want to spin our samples at higher frequencies so that we can more effectively average out the interactions in the Hamiltonian, especially such as the homonuclear proton-proton interaction. All of these together, I tried to put this on one slide, makes for happy NMR, happy spectroscopists, and high impact field. So higher magnetic fields, we need stronger magnets. We're not only implementing commercial magnets that we buy from Bruker, we're also building our own magnets at 28 Tesla and higher now. For the microwave pulses, we're generating these pulses with custom built gyrotrons. This will allow us to do pulse DNP transfers. Our main goal of pulse DNP is to perform experiments at room temperature or higher, and also at very high magnetic fields efficiently. And to access even higher spinning frequencies, we're going to be implementing these spherical rotors, which will also enable better access to temperatures below 4 Kelvin. We'd actually like to spin in the superfluid. I have a disclosure that some of this technology is already patented at Washington University in St. Louis, and we are in the process of making an ET8 spinoff to also commercialize most of the technology you will see today. So how do we do pulse DNP at 1200 megahertz and higher? We need to have knowledge and understanding of the magnetic resonance Hamiltonian, and we need to be able to control the internal Hamiltonian with an external Hamiltonian. So the magnetic resonance Hamiltonian is of course dominated by the Zeeman interaction. At a magnetic field of 28 Tesla, that Zeeman interaction on the proton spins is 1200 megahertz. That's 792 gigahertz on the electron spins with a G factor of about 2. So we can control that Zeeman interaction by building and using high field magnets. There's a chemical shift anisotropy that's maybe about 40 kilohertz. Of course, this depends on the field and on the spin of interest. But again, we can control this chemical shift anisotropy by having high spinning frequency to partially average out the anisotropy, and also radio frequency pulses to control it. The dipolar interaction we need to have knowledge of and control of, not only to, for instance, measure those dipolar couplings that would give you structural information if you want to determine the structure of, let's say, a drug when it's binding to a protein, but also dipolar couplings allow you to transfer polarization in the cases of DNP and cross-polarization, but you need to be able to control the dipolar coupling so you can do proton decoupling, so you can do electron spin decoupling. So we control the dipolar coupling with an external Hamiltonian as well. We also need to be aware of the scalar interaction. There's only about 30 hertz. We can control this mostly by spin dilution, and we can also have radio frequency decoupling. So that here are the internal interactions. We control these internal interactions with an external interaction. For carbon and nitrogen spins, we have about 100 kilohertz on the probes that we build in our laboratory. The protons, we're also at about 100 kilohertz right now, but we're building new NMR probes, and our target is 2 or 3 megahertz for gamma B1 on the Rabi frequencies to better enable pulsed DNP transfers. On the electron spins, the interactions are really strong in the Hamiltonian, and we don't have control over them yet. All right, so the internal interaction on the electron spins is far larger than the external interaction we can presently generate in match angle spinning. This is the main challenge of our field right now to perform pulse DNP. So right now we have about a megahertz, maybe up to 2 megahertz gamma B1 or Rabi frequency on the electron spins. But if you have more microwave power, if you focus the microwave power with lenses, let's say, and even if you have a resonance structure, then you could get 20 or 30 megahertz gamma B1. If we had 20 or 30 megahertz gamma B1 on the electron spins, and we could generate pulses, we can do pulsed DNP transfers. Here are the magnetic fields that have progressed over the decades. This data is from Maeda et al. Last year in JMR. And you can see there's a slow but steady increase in NMR magnetic fields, which enables more higher impact experiments. Here are 3 pictures of high field magnets that we will have at the ETH in the summer after we receive our 1.2 gigahertz. In our laboratory, we want to get to even higher magnetic fields. And these new Rebco tapes, type 2 superconductors, we think will enable that. So in our laboratory now, we are in the process of designing new magnets that we hope will enable access to much higher NMR magnetic fields. So we're already in the process of designing a 28 Tesla magnet, and we're thinking about designs that could be implemented at 50 all the way up to 100 Tesla. You can only do this because we have this new Rebco tape, and there's been a huge amount of technology development in these Rebco tapes lately. So the magnet has the superconducting tape. We wrap it up in these pancake coils is going to be the plan. This follows very closely to the papers and pioneering research by Yuki Iwasa and colleagues just down the hall from Bob Griffin and Kong at the Bitter Magnet Lab. And we've done magnetic field analysis that shows that we should be able to get to a field of about 29 Tesla. We also are doing a lot of stresses and force analysis. That's one of the biggest challenges with designing superconducting magnet. And we're already in the process of fabricating and starting with these magnets. So mind you, I've never built a magnet before. Not sure how many people are on the Zoom call have. I know that Bob has built a magnet, that might be it. We have some experience with engineering, but we're not going to let a small thing like not having any experience with magnet technology prevent us from building new magnets that we need to push the field forward. So this first 29 Tesla magnet, our plan is to build it just within our group. I gotta tell you, we're almost like purposely not looking at magnet designs because we want to make sure that we're designing this magnet from first principles and we can approach the magnet design in a new way. This first magnet will be for a first harmonic gyrotron in order to generate 1 kilowatt pulses for pulse DNP at 792 gigahertz. And with the gyrotron magnets, they are way easier than an NMR magnet. You can run them in non-persistent mode, so you don't even need a superconducting joint. There's a very moderate field homogeneity. I'm talking like 1% homogeneity is enough for a gyrotron, not something like 0.1, 0.01 ppm. However, in the future, the idea is to use this first magnet as a stepping stone to indeed making NMR magnets that are very stable, higher, even higher fields up to 100 Tesla, and have the sufficient homogeneity to do NMR experiments. Those are for pulse DNP experiments at really high field. Right now, we're doing continuous wave DNP experiments at 7 Tesla, and we follow the pioneering research by Bob Griffin and his colleagues over the years at MIT that have developed these cross-effect mechanisms and radicals. So we use Amupol in our case. It was actually introduced by Lyndon Elmsley and colleagues. And these cross-effect radicals, you can saturate one of the electron spins with continuous wave microwaves. When that electron spin transition is saturated, it wants to reestablish equilibrium, and it does that by flipping a second electron spin and also an NMR spin. And we can get really high sensitivity, really high DNP enhancements in our hands of about a factor of 280 on model systems, and this is done at a temperature of 6 Kelvin. So together, this should give you a theoretical enhancement of about a factor of 11,000. However, these experiments are really cold, cryogenic environments, And for that, we build cryogenic sample ejection system, so we don't have to warm up our entire probe. We can simply spit out our sample and drop it back in the magnet. This really increases efficiency of our experiments. However, these cross-effect DNP experiments, as described by Tycho, by Robert Tycho, by Griffin, by Gail DePep, and many others, you could have a lot of paramagnetic quenching and depolarization, depending on what radicals you can use. As those groups have also pioneered, you can use nice creative biradicals with tritol to get around this. Nonetheless, we feel like the best way to really overcome paramagnetic broadening and even depolarization is to use narrow line radicals and control those electron spins. Okay, we want to control the internal interactions of the electron spins using strong microwave fields. The stronger the microwave field, the better control, the more pulse DNP and electron decoupling we'll be able to do. So Sabrina Chen in the lab has designed these Teflon lenses that focus the microwave power. It's very similar to the optical lenses that focus microwave power into very small spheres. Why are we using spheres? Well, we want to focus all the microwave power as possible, and that means the cross-section area of the tightest beam you can focus an electromagnetic radiation into is a circle. That means inherently, if you want to get the most bang for your buck for microwave power, you want to use a spherical rotor. With 5 watts of microwave power incident, we calculate using a high-frequency structure simulator that just solves the Maxwell equations. We should be able to get about 2.5 megahertz gamma B1 with 5 watts of microwave power. With the new Gyrotron oscillators we're building that can give us more than 500 watts, we expect to get to about 20 megahertz gamma B1, which we believe will enable pulse DNP applications. Not only do we want to do pulse DNP transfers, we also want to continue decoupling the electron spins. What do I mean by that? I mean, we want to transfer the polarization from electron spins to carbon spins. We can also do this to proton spins. And then when we acquire the carbon-free induction decay, we're doing this not only under proton spin decoupling, but also under electron spin decoupling. So in this case, we can decouple these tritol electron spins. The effect of electron spin decoupling is very similar to the effect of proton spin decoupling. The resonances that you observe become more intense and the line widths become more narrow. And this works in model systems. We also can decouple tritols with inside bacterial cell lysates. And now recently, we've also demonstrated that we can decouple a different type of electron spin, specifically an endofullerene. So that's an atomic nitrogen within a fullerene cage. We really think that these fullerenes are going to be important in the future because they have very narrow line widths. This G anisotropy is almost not existent. This means that the resonances of the electron spin are very narrow. They're about 1 megahertz broad. This is discussed a lot more in some beautiful papers by Bjorn Kurzelius et al. If you only have 1 megahertz line width, that means you only need a 1 megahertz gamma B1 field to control those electron spins. That is going to better enable pulse DNP in the future. The fullerenes also, this EPR spin is kind of protected, so it doesn't react with your sample, which is another major advantage. So how does electron decoupling work? We've done the spin physics to simulate this, as pioneered by Eddie Saliba and also Michael Mardini. Have done calculations, and the idea is that we make an adiabatic chirp over the EPR resonance. We chirp back and forth, and this takes the electron spins from spin up to spin down to spin up to spin down. We continue these chirps, and if you can get the electron spin to go spin up to spin down faster than the hyperfine interaction takes to evolve, then you should partially average that hyperfine interaction, hence electron spin decoupling. We can do continuous wave electron spin decoupling, where we just park that microwave frequency on the top of the EPR resonance, and we see we can improve the line width by about 15 hertz on our model system of interest. But if we do these chirps in the microwave frequency across the EPR line shape, we get much more effective decoupling of the electron spins, up to about 35 hertz in this model system looking at a bulk sample. We've taken this to the next level. Nick Alaniva, Eddie Saliba et al. Published in Agabanta, where we hone in and we look at very close nuclear spins, very close to the electron spin. So the idea is now we can transfer polarization from the electron spin to these first directly coupled nuclear spins. Typically in DNP, you transfer polarization to a ring of nuclei, and then you rely on nuclear spin diffusion to pump that polarization to the bulk. This is different. In this case, we're looking at directly hyperfine coupled nuclear spins. And without electron decoupling, they're almost totally washed out, shown in black. When we turn on electron decoupling, we get a massive gain in sensitivity. And we're only allowing the DNP to be on for 5 milliseconds. So we do 5 milliseconds of DNP, then we acquire a signal. So we can almost get about 20% duty cycle of observed FID. So Yoshitaka Ishii, one of the many heroes I have in the field, he likes to say, all those beautiful silver NMR magnets we have, they're just sitting there collecting dust 98% of the time. And he's totally right, because you're just waiting for proton T1 and waiting, 3 seconds, and then you require maybe 50 milliseconds if you're lucky of an FID. Here, what we're proposing is that we instead do a direct transfer from the electron spin, and then you only have to wait for the electron T1. And the electron T1 is much shorter. So for the first time using this technology, we've been able to measure what the electron T1 is in match angle spinning at 6 Kelvin in this case, about 6 Kelvin. We can see that the electron T1 is about 34 milliseconds. So in these experiments, we're doing electron decoupling over the FID. This is effectively saturating the electron spin resonance. Note, the electron saturation is not a mechanism of electron decoupling. It's a side effect of doing the chirps across the EPR resonance. The mechanism of electron decoupling is flipping these electron spins back and forth faster than the hyperfine interaction. Nonetheless, you have a saturation on the electron spins, and then you can do a saturation recovery just like you do in NMR. So we turn off the microwaves using our new gyrotron we've built for a period that changes from 0 to 100 milliseconds, and then we do 5 milliseconds of DNP, and then we can measure a T1 saturation recovery experiment on the electron spins, indirectly observing the carbon spins. This electron decoupling is very broadly applicable. We've done this now within intact human cells. We see a similar gain in intensity for the bulk using, this is a longer relaxation, a longer recycle delay of about 10 or 20% inside these human intact cells. So there's 12 million Jurkat cells in our 3.2 millimeter rotor spinning at 6 Kelvin. Another very interesting result we saw when we have these tridal radicals inside these blood cancer cells, or the lymphocytes in this case, is that the proton T1s, and this is bulk now, this is not electron T1, this is doing DNP like it's usually done in MAS. So we polarize proton spins, we have proton-proton spin diffusion that diffuses out. That buildup time is only 300 milliseconds at 6 Kelvin when we're spinning, when we use these special new radicals that are designed and synthesized in the group of Snorri Sigurdsson. They have these other methyl groups on them. 300 milliseconds is all the T1 is, even at a temperature of 6 Kelvin. So not only does DNP give you huge gains in sensitivity, you can actually repeat the experiment faster with DNP than you can conventionally, even at a temperature below 6 Kelvin. Why are we doing these experiments within intact cells? Well, the cellular context is the most relevant. It's the most realistic environment to examine proteins. That's where proteins work. That's where amyloid fibrils form. That's where drugs interact with proteins. We want to examine the proteins, the machines that we're looking at in their endogenous context. We also want to be able to directly correlate this structure that we determine with NMR to the phenotype. So in this case, our application of interest is to activate latent HIV reservoirs. So here you can see an assay we've performed in our lab. If these cells are green, it means that the cells have turned on. So what we'd like to do is be able to determine structures of drugs, let's say, and then also look at the phenotype that you're interested in eliciting from that drug in the same exact sample. So you have the most relevance. It's also a lot easier to make human cells and bacteria cells for a sample. So we don't want to spend a year optimizing the protocols every time we look at a new protein. In-cell NMR is great. I need to mention that, of course, it's been pioneered by many people in the field, such as Selenko, Roland Reek, and many others. All you need to do, in-cell NMR at a totally endogenous concentration, not overpopulating or overexpressing those proteins of interest, all you need to do is get about 1 million times higher NMR signal. So that's exactly what our plan is. So we're going to get that NMR signal increase by using dynamic nuclear polarization, going to lower temperatures, and also going to higher magnetic fields. So here, rather than using tridal and solid effect radicals, we use a biradical, again, Amupol. We put it inside our intact human cells. That Amupol goes right through the membrane on intact cells, human cells, the mammalian cells. This isn't the case in bacteria cells, but you don't need to add anything special to the radical. It goes straight in. You do need to be careful that these radicals can get reduced inside the environment of the human cell. So our solution to that was just to work quickly. So Bryce and Chukun, when they make these samples, you add the radicals and then they diffuse through the membrane very quickly. And then you get that sample, centrifuge it down into the rotor, freeze in liquid nitrogen, all within about a minute, and then you can do your experiment. Again, the T1s that we see are way shorter than we were expecting. The T1 is only 1.7 seconds, 1.26 times T1. The optimal recovery delay is only about 2.5 or 3 seconds between these experiments. We got large DNP enhancements using Amupol of about a factor of 50. When we first did this, we didn't even really believe these experiments could be working so well. I need to add also that these experiments are done at a temperature of 5 Kelvin, so the effective sensitivity gain is around a factor of 2,000, although there is some depolarization present. So we weren't even sure that these experiments were working as well as they looked like. So we made a new polarizing agent that has a fluorescein on it. All right, so the idea here is to optically localize where that polarizing agent is going inside the intact cell. So we have a biradical, then we have a fluorescein, and then we can optically locate where this trimodal polarizing agent is going within the intact cell. In this case, we also use a TAT peptide, which is targeting this whole trimodal polarizing agent, in this case to the nucleoli, but you could use any targeting domain you want. Our vision for this is that you could send DNP radicals to the mitochondria, to the cytosol, to the Golgi, whatever organelle you're interested in, and also examine the samples at the same time with a fluorescent or optical microscope. So again, in green, this is where the radical is going. So indeed, yes, these DNP polarizations enhancements we're seeing are coming from intact human cells. Another advantage to DNP within intact cells is that you can combine DNP and NMR with other molecular biology techniques, such as using other dyes. So here in blue is DAPI, that goes to the nucleolus. So you know where the nucleolus is, you know where your polarizing agent is in green. And you can even combine this also with other molecular biology techniques, such as flow cytometry. So as we move more into using intact cells for our samples, it's going to become more of an issue and an opportunity that we are actually looking at the heterogeneous population of the cells. Even if you're looking at clones like we are usually in our laboratory, there's still a big heterogeneity in the cell population with regards to transcription levels and cell cycle, et cetera. So in this case, all of the cells don't behave the same, but flow cytometry lets you look at each individual cell at a time. We can analyze each individual cell's ability to activate these latent HIV reservoirs with different drugs, in this case, tumor necrosis factor alpha, bryostatin, and a control of DMSO. And now we can correlate that to the amide backbone, which is also showing you protein expression levels. So for the first time now within intact cells, we've been able to use DNP enhanced NMR to determine the activation level of latent HIV reservoirs. It's done by Dr. Sarah Overall and Lauren Price in the group. In the future, we'd like to combine these experiments with intact cells with pulse DNP at 37 degrees Celsius. Why do we want to do pulse DNP at 37 Celsius? The main reason is that proteins are moving. We want to be able to determine that molecular motion experimentally with DNP, with NMR. And there's not much motion when you cryogenically freeze everything out. Hence, we need to do pulse DNP at 37 degrees Celsius. We also expect a much better improvement in resolution with DNP. So why is it that DNP spectroscopists are usually the only ones that are doing cryogenic MAS? It's because for dynamic proteins that are sampling a lot of different structures inside their conformational ensemble, typically the lines are broad. All right. Now, this isn't always the case. When I was a graduate student with Bob, we looked at a couple of systems, including crystalline peptides like MLF and also bacteriorhodopsin, and we see pretty narrow resonances. Okay, we got 30 hertz on crystalline MLF in cryogenic MAS, and we got about 1 ppm on bacteriorhodopsin, the active site. Of course, there have been other really nice studies by Adam Longa and Anne Lesage and many others lately that show that when you have some systems that are not sampling a lot of dynamic conformations, then you can get narrow resonances cryogenic with DNP. But typically, if something's moving around, you freeze out all those different conformations and it can give you broad resonances. That's why we want to do pulse DNP at room temperature so we get high resolution spectra and at high field. Our strategy is to use pulse slicing this is following strategies from UC Santa Barbara, or frequency agile gyrotrons to make chirped microwave pulses and hard pulses to do coherent transfers of polarization using strong hyperfine interactions that allow for fast transfers of magnetization from electron spins to nuclear spins. If you use a strong hyperfine interaction to transfer the polarization faster than the electron relaxation time at room temperature, then typically you're just going to have a really broad spectrum on the nuclear spins. So you need to combine these pulse DNP experiments with electron decoupling during the acquisition to regain high resolution spectra. In order to do this, we're going to need new microwave sources. We've developed frequency agile gyrotrons in our group. Here you can see how we can tune the microwaves over about 100 megahertz in our gyrotrons. Our gyrotrons have an emitter at the bottom of the gyrotron that's barium impregnated tungsten. If you heat this tungsten to about 600 degrees Celsius, it'll eject electrons because it also has a really strong negative potential. Those electrons will feel that potential and they'll be ejected orthogonal to the surface of the cathode. And then the gyrotron is also in a really strong magnetic field. So that's an E cross B, that's a Lorentz force. So the electrons start spiraling up. They go into the interaction cavity of the gyrotron, at which point they're moving, accelerating charged particles, right? We all know from the Bohr theory of the atom that accelerating charged particles have to give off energy, right? This is how gyrotrons work. So gyrotrons give off energy when the electrons are going through their cyclotron resonance motion, and the energy that they give off is the microwave power that's emitted from the device. So this microwave power is emitted in this TE5-2 mode. The electron beam resides on the first radial maxima to get good coupling with that interaction cavity. And then we extract the microwave power with an internal mode converter that comes out of sapphire or diamond window. How do we make the pulses, the chirped pulses? How do we get frequency agility? Well, as follows: if you can just change the potential of this copper piece over here, this anode, then you're changing that initial acceleration of the electrons. And then that means you're changing the cyclotron resonance motion and hence the microwave frequency. So that's exactly what we do. This voltage now is integrated to the spectrometer of the NMR via an arbitrary waveform generator and a high voltage amplifier. And this allows us to make these chirps of the microwave power. This is pioneered by Faith Scott, now a postdoc in Florida, and also Chukun Gao in the group. We build these gyrotrons ourselves. Here you can see students from the group assembling the gyrotron for a high temperature bakeout. These are vacuum electron devices, so you got to have a really low pressure in order to make sure that that electron beam doesn't fizzle out and you don't get adverse chemical reactions on the surface of the cathode. So as assembly of the gyrotron, we get very continuous tuning of the microwave power as we change the voltage. This is important for a chirp. And in this gyrotron, we were able to get about 10 watts of power over about a 1 gigahertz tuning range. Our new gyrotrons give us about 50 watts over a broadband. We use these new gyrotrons we've built to perform chirped DNP with Magic Angle spinning. So in this case, we used trityl nitroxide biradicals. Eddie Saliba in the group did some spin physics calculations to show here's a narrow trityl resonance, here's a broad nitroxide resonance, and we make this chirp just over the trityl. There are also very similar elegant experiments coming out of Song Yi Han's group from UCSB. I expect that you'll hear some of these next week. In this case, we're using a chirped gyrotron, and we get about a 20% gain when we make these chirps over the solid effect condition. So we get an enhancement going from 115 to about 137. The faster we do the chirps back and forth over this condition, the better we get for the enhancement. And so we're making new technology now that can get to even higher enhancements using even faster chirps. But the chirped DNP is really just a transition in our case to pulse DNP. Okay, so these experiments that I showed you are all custom. So the microwaves come down this corrugated waveguide, they shine on the rotor that's spinning. We then have a radio frequency coil surrounding our sample to detect and manipulate the nuclear spins. We've done a computational fluid dynamics simulation that shows you all of the cooling and the heating from the spinning gases. Here's the bearing, here's the drive, here's a VT gas that comes in and blows on the rotor. In this case, we're using helium, liquid helium, that comes down this vacuum jacketed line and blows on the center of the sample. And we can do these experiments at below 6 Kelvin, spinning up to a spinning frequency of about 6 kilohertz, but it costs about 40 liters of liquid helium per hour. That's a lot. So can we improve that? You know, there's some other things that we'd like to improve in the technology. We'd like to spin at higher frequencies. So I'm really inspired by many pioneers in our field that are spinning at higher and higher frequencies to average out proton-proton interactions, to increase proton resolution, giving us better sensitivity experiments. And those experiments are mostly done, you know, above 40 kilohertz, 80 kilohertz. The record, as far as I know, is about 160 kilohertz coming out of Bayat Meyer's group here at the ETH. And we want to also do DNP at the highest spinning frequencies possible. And with these cylinders, it's hard. Another disadvantage of these cylinders and our setup as we have it now is that you have this coil, which is blocking the microwaves from shining on our sample. So even if you pick the spacings better to transmit the microwaves, which can be done, they still will interfere with your ability to convert linear polarization to circular polarization and maintain that circular polarization, which gives you a higher gamma B1 field. This is brought up in a nice paper by Tico and Thurber. Okay, so these are the improvements. And the last thing that has been really frustrating me with magic angle spinning technology the last few years is this sample eject. All right, so this is the first project I did with Bob in graduate school. I designed a sample eject that turns this rotor from the magic angle to vertical. And I've spent like, you know, 10% of my time in my career building cryogenic sample ejects. I'm sick of it. I never want to do it again for cylinders. So we thought these are all the challenges. You know, we want better microwave coupling for pulse DNP and higher gamma B1s. We want faster spinning frequency, lower temperatures, better sample ejects so that you can do these experiments in really narrow bore magnets. That's important to get to higher fields. And our inspiration came from nature. So we thought, you know, how does nature spin samples? It's always a sphere, the Earth, a pulsar, the sun. So we started spinning spheres. Here's a 9.5 millimeter zirconia sphere. Spinning very stably at the magic angle. So our strategy here is to get zirconia spheres. These things are actually sold as industrial lubricants, yttria stabilized zirconia. This is the same material that most everybody uses for zirconia for cylinders for magic angle spinning. You buy these spheres, you drill a hole in the middle, and you pack your sample, which is almost always lower density, samples lower density than the zirconia. Then you have a stable, favorable moment of inertia. So these rotating objects always want to put the higher density on the outside of the rotation. This gives us very stable spinning about the magic angle. Another way to say this is that once the rotor spins at the magic angle, you know, you saw that it was a bit rocky to get started, but once it's stable, once it's spinning, because this isn't really a true isotropic sphere, once it's spinning, if you were to wobble off the magic angle, that would cost you a lot of momentum, right? You really want to conserve angular momentum, and this is why magic angle spinning spheres are so stable on their angle when they spin. Another big advantage of these spheres is they're much more mechanically stable. So where the most stress is on these spheres is where the sphere is the thickest. So we've performed over, 1,300 different stators, thousands of different experiments over the last 1 or 2 years, and we've only broken 2 rotors, 2 spherical rotors over that time. In the first year of my laboratory, we probably broke about 30 sapphire and zirconia rotors that were cylindrical. So how do we do this spinning of the sphere? We have one aperture of gas that holds up the rotor, this brainchild of Sabrina Chen, that holds up the rotor, and it's offset at the complement to the magic angle. Then you'll get spinning about the magic angle. So whereas cylinders, you have to have, 7 or 8 holes on each bearing, and you need to have all these complicated holes on the turbine, and then a VT. It's all very complicated. Spheres are really easy. You just need one aperture that holds up the rotor, supplies turbine energy, and also variable temperature gas for thermostating control. We make these stators with 3D printing technology, both in plastic and also in ceramic. This 3D printing allows us to fail fast, fail often. So we've made more than 1,000 different versions of these stators over the last 1.5 years, which has allowed us to do spinning on these samples for 9.5 millimeter rotors, and now also 4 and 2 millimeter rotors. So again, you can see that the sample ejection is now very easy. So you get this rotor, and you just have to have a tube that comes in from the top of the magnet, and you can just suck out the rotor. This is going to allow you to shrink the whole magnet board to much smaller diameters. And this is why we think we're going to be able to get to 100 Tesla. So the angle is very stable. This is not one shot of KBR. It's an average of 256. And you don't see spinning sidebands that go out to 10 milliseconds if you don't have stable match angle spinning about 54.7 degrees. So indeed, the spinning is very stable on the metric angle. The spinning frequency is also stable. Chukun Gao implemented this resistive MAS regulation introduced by Terry Gillian, and she gets plus or minus 1 hertz of spinning frequency stability on the spheres. It's actually probably better than that. That's just our ability to do spinning detection. And with helium gas, we can spin the 9.5 millimeter spheres at up to 10.5 kilohertz. So that's more than twice the frequency you can access with 9.5 millimeter cylindrical rotors. Dr. Tom Popp in the group, our new postdoc, has made better turbine grooves on these rotors that can kind of catch the air a little bit better to get better coupling of the turbine gas to the rotor for better spinning. And he thought when he did these different rotors that he'd have a control, which is with no grooves. And I told him I thought that was a bad idea. It's never going to spin. Turns out controls in science are a good idea. You should always have a control. And Tom's control rotor actually spun at 4.3 kilohertz and stable on the magic angle with no grooves at all. This shows you that the stable spinning on the magic angle is not because the gas is grabbing the grooves. It's because you have a stable moment of inertia and you're putting that higher density zirconia on the outside of the equator while it's spinning. In this case, you're getting the turbine energy just from the friction of the gas sliding over the surface of the rotor. We've been able to make these stators now in ceramic with 3D printing, which is going to be really important for reproducibility and commercialization, and also our ability to use these systems for cryogenic applications with DNP. Tom was able to, within about 2 days, print these new stators on the 3D printer that was already at ETH Zurich. I think he still had jet lag when he got this experiment to work. And he got a commercial probe that was hanging around Meyer's lab, not in use, and he was able to convert the commercial probe to magic angle spinning with spheres in about 2 days worth of time. We've been able to scale this technology down to 4 millimeters, which allows us to generate high intensity radio frequency fields in order to do proton decoupling and also cross polarization experiments. And now we're using these double saddle coils. So I had a very informative talk with Rachel Martin and UC Irvine. I asked her, Rachel, what in the world should we be using for these inductors for the spheres? I mean, we have these split coils and they're really bad coupling. And she said, hey, we should really look at these double saddle coils. So her graduate student, Jessica Kautz, gave us the CAD design for these double saddle coils, which really has a big improvement on our filling factor of the spheres and our radio frequency coupling. So here you can see a video of the 4 millimeter sphere. And what you should be looking for is, as it spins up, this black dot on the end is kind of coming into focus. And that's because you're establishing a more stable spinning axis about the magic angle. Lately in our group, we're now scaling this down to 2 millimeters. This is pioneered by Lauren, Chukun, and Sabrina in the group. And just recently now, we can spin the 2 millimeter spheres at a magic angle spinning frequency of about 55 kilohertz. So again, the spheres are always doing almost twice as good, or factor 1.6 as good as the cylinders. This is why we think we're gonna be able to access much higher spinning frequencies with the spheres. We're also in the process of scaling these down to not only 0.5 millimeters, but Chukun Gao, postdoc in the group, just put in a purchase order for 0.2 millimeter spheres that we are hoping we're gonna be able to spin well north of 200 or 300 kilohertz. So the idea is that if you make a really small sphere, you'll be able to focus all of that microwave power into a tiny point to do pulse DNP at room temperature. You'll be able to do really high spinning frequency, so you can do proton detection. We'd even like to do electron detection. So as the spheres get to 0.2 millimeters, you might think, you're not gonna have any sample left. Where's the signal gonna be? And I'm inspired by a lot of the pioneers in our field that have shown as you go to smaller diameter cylindrical rotors, you can spin at higher frequencies. That enables you to detect on a higher gamma nucleus like protons, and you recover that sensitivity back. Well, we can imagine if you can recover that sensitivity back by spinning electron spins, you're going to get much higher sensitivity. And that's presented in a paper published by Eddie Saliba in Journal of Chemical Physics last year. With that, I'd like to thank again everybody in my group. I mentioned a lot of them along the way. I don't think I did mention Lynn Farner and Alexander Dapp and Ronnie, who are making these new magnets. Patrick Judge, he's doing the structural biology with us. Shannon is making proteins with us. Here are our new students that are going to be implementing this technology. The tridal radicals are synthesized by Snorri and Thomas in Iceland. We currently design our gyrotrons in collaboration with Jags Sirigiri at Bridge 12. We look forward to continue working with Matthias at the ETH, along with all of our other fantastic NMR collaborators at the ETH, continuing our collaborations at WashU with Sophia and George and Paul Wender at Stanford. And I'd like to really thank the previous members that really did a lot of the research I showed you today. Eddie is a postdoc at MIT with Bob. Faith is now at the High Field Magnet Lab. Bryce is in Cincinnati. Michael and Natalie are also graduate students with Bob. And I'd like to thank the ETH and the NIH for funding. And I'd really like to thank everybody for your attention today. As I mentioned, and as Kong told you, I have the slides for all of this presentation available on Dropbox. So please visit that if possible. And I'd really like to thank you guys again for your attention. I'd like to thank Bob and Kong again for putting this on. I think it's going to be an important aspect of keeping our field together. Thank you.
All right, thank you, Xander, for your very, very interesting talk, very, very inspiring, very new science, and I find it very exciting. So just for all the participants, I know that you cannot see other participants, but let me tell you, we have 250 people on now. So 250 people looking at this, and I'm going through a list, there are so many people. And so I will now proceed with the Q&A sessions. If you open the Q&A, I can see there are already 13 questions. And then with, those, as I say, while I'm talking about this, you can also type in your question, and those with the most votes, I will get asked first. So I will first proceed by taking several questions from Q&A, and at the same time, those who would like to ask questions verbally, orally, you can raise your hand, and then I will unmute you, and then you can speak. So I will first go with the first question that has 12 likes. So someone asked, how is the RF inhomogeneity inside a sphere rotor compared to that inside a cylindrical rotor? And someone commented on it. How does the filling factor, if you compare these two, sphere and cylindrical rotor?
Okay, so thank you. It's a very good question. The radio frequency homogeneity actually isn't that bad. It's comparable to what you get on using a solenoid or even a variable pitch solenoid. However, and so we've done this radio frequency analysis. We are going to be publishing a paper about this soon. The filling factor is really what is a little bit more of a challenge for us right now. So the filling factor is slightly worse on the spheres at the moment, but we have ideas in which we can kind of continue to bring that saddle coil closer and closer into the stator. So Tom Pop and Chu Kung Gao and Sabrina in the lab actually have designs that kind of combine the stator with the coil to improve the filling factor. So we actually think that we're going to be able to get something that's comparable with the cylinders, partially because remember with the cylinder at the magic angle, you're not trying to beat a solenoid that's perpendicular to the field. You already have this cosine 54.7 degree loss of coupling to detecting that transverse magnetization. So we're actually optimistic that we will be able to get a better filling factor, but we're doing the radio frequency analysis. Okay, so let me be clear. I think we're going to be able to get a better filling factor. However, I will say, if you got to pick between having a slightly less filling factor, but 100 times better gamma B1 field on the electron spins and spinning at 500 kilohertz, well, I'd probably rather pick that.
All right, thank you. Just to let you know, like in the Q&A function, there's an option to stay anonymous. Like if you don't click anonymous, then I will read your name, like if you don't mind. So like the next question with 13 likes, also quite a lot of like people like this question. So David Bryce asked, electron decoupling provides a gain in signal to noise ratio, but does not seem to narrow the resonances. Can you comment on this?
So when you do a detailed analysis, it actually does narrow the resonances. This is not only in terms of T2, but also homogeneous T2. So we've done CPMG echo trains, and we do have a longer homogeneous relaxation time. This is published in the original JAX paper. However, you're absolutely right, especially for the Angevanta paper where we looked at those carbon spins that are directly coupled. We don't see actually any difference in that line width. And we think that that's because, to a large degree, those carbon spins that are very close to the electron spin are so horribly broadened that we don't even see those signals at all. So the electron decoupling just kind of brings those back into the fold and you can see them again. However, you know, these are just the first experiments, okay? You know, we think that it's going to take years to really develop electron decoupling to the point where it's as sophisticated as proton decoupling. You know, for instance, these chirps aren't where we want to be in the future, okay? How do you want to do electron decoupling in the future? You want to have a fullerene that's only about 1 megahertz broad, and you want to hit it with pulses. You want to have like TPPM, you know, or XIX on the fullerene. At that point, you'll really be able to see, I think, these improvements in the relaxation time and the T2s and the line widths. But in order to do that, you need to have better gyrotrons, and you need to be able to build these gyrotrons at higher magnetic fields.
Okay, and the next question, someone asked, 2 millimeter spheres spun at 55 kilohertz speed exceed the speed of sound at the largest circumference. Is that not causing problems? And someone say, is it using a cold helium gas to spin it?
So that is using helium gas, and our calculations show that that is not exceeding the speed of sound of helium. So that's actually not a limit for us right now. We're at, I think actually in helium, that's only 0.4 or 0.5 Mach, 0.6. So we can maybe talk about that more. Normally at a conference, I would say over a beer, but that would be a morning beer for you guys. Maybe you don't want to do that. But by our calculations, we're only at about a half a Mach.
Okay, would the spheres be at all compatible with samples with densities greater than that of zirconia, which is 5.7 gram per cm cube?
Ooh, that's a tough one. Yeah, I could imagine, I hadn't thought about that before, so thanks for the question. I could imagine how you could design the sphere. So for example, so that the sample is further out on the equator and you have zirconia and other places of the rotor. So you can actually, we're trying to 3D print the rotors themselves. So if you can 3D print the rotor, you can pick whatever geometry you want to establish whatever moment of inertia you want, depending on the different densities of the sample. So that's a good question I haven't considered, but absolutely, I think we could do that.
Okay, and then next question from Songi, and she asked, what DNP mechanism would you rely on for DNP at room temperature? Ultimately, the electron-electron and electron-electron nuclei dipole coupling will be averaged out. So it cannot be traditional cross effect, solid effect, or turbo mixing DNP effect. Any thoughts on this?
Right. Yeah, so thank you, Songi, for the question. Our favorite pulse DNP mechanisms or chirp DNP mechanisms are, following Kong's research. So you could answer that as well as I can almost, Kong. I mean, we like the integrated solid effect a lot. We like the adiabatic sweeps that Kong and Bob and Matthews and many others in Bob's group have done. The off resonance adiabatic sweeps through the novel condition, I think, are really, you know, exciting. If we could implement Kong's top DNP, that would be exciting as well. You know, lately, Song-Yi, we really like electron nuclear cross polarization. So turns out CP, that's what we've been using for years in our field. And if we can get the CP Hartmann-Hahn condition, this is why we want to make the proton gamma B1 at like, you know, 2 megahertz or something ridiculous like that. But if you make the rotors 300 microns, yeah, you know, that's getting achievable. If it's that small, we think that we can generate 2 megahertz on the proton spins. And then you need, you know, 2 megahertz on the electron spins. We're basically already there. And then we would want to use something like electron nuclear cross polarization. And in that way, then the match angles will do cross polarization and use those strong dipolar couplings from the strong hyperfine interactions to do the transfer faster than electron T1 at room temperature. The last thing I'll add to that is like, but you really want to use the longest T1 possible on the electron spins. And this is also why we're really excited about these endofullerenes. We want the longest possible homogeneous electron spin at room temperature. And tridal, or especially the fullerenes, we think are the way to go.
Yeah, I just want to like comment a little bit. I mean, it's interesting, like if you like spin so fast, then when the spinning frequency, it becomes like comparable to the size of your dipole coupling. I think we are back to what we are very, very good at doing solid state NMR. You can do the recoupling experiment, you know, reintroduce it back. And also apart from that, what we are doing across effect, solid effect, you are using the pseudo-sacral hyperfine coupling B, but there's actually also the isotropic part and the AGG part if you want to do solution state pulse DNP in the future. I mean, this is something.
Yeah, how about,
well, we can talk about this.
How about electron nuclear TROSY or electron nuclear TDOR? I mean, this is what we're trying to do with the field. We're trying to get control over those electron spins so we can just go to the amazing literature that's been out there for the last 40 years in NMR and do the same thing on the electron spins. We just need the technology to get there. Yeah,
we need the hardware from you. So we'll talk about this next time. So I want to attend more questions first. All right, I know it's getting exciting, but all right. So someone asked, how do the cells behave when spun at such high frequencies?
So my postdoc, Sarah, overall has done these experiments at room temperature when you spin bacteria cells at room temperature. And it's kind of what you'd expect is what she tells me. They die right away. So in our experiments, it's very important that these human cells that we're looking at are frozen. Okay, so we freeze the cells actually in about 10% DMSO. So we use the same cryoprotectant that we use when we store the human cells to keep them safe in the freezer at -80. So it's about 10% DMSO, and the cells are already frozen. So whatever state they're in at room temperature, you cryogenically freeze them, and once they're frozen, they're not going to change when you start spinning them, we think.
Okay.
Yep.
All right. So the next question from Dominic, and he asked, like, how precise can you control the magic spinning angle?
Okay. So the precision, I think the best answer to that is that right now on KVR, we can get those spinning sidebands out to 10 milliseconds. You're probably asking, well, why don't you do some ST-MAS to actually figure out what that angle is? We want to do that also. We haven't done it yet. So all I can tell you is what we've seen on KVR. We're getting to probably better than, I don't know, I want to say 0.1 degrees better than that. But we need to do an ST-MAS experiment to answer that question, and those are ongoing.
Okay. Next question from Oliver Lafont. What is the broad size of the 29 Tesla gyrotron magnet you plan to build? And what are the limitations for going to higher than 50 Tesla?
15 millimeters. So we're going to make a really tiny gyrotron. That's how we're gonna actually build these 29 Tesla magnets in our own lab. So we're going to try to fit the whole gyrotron, and that's like the inner bore size of the magnet. That doesn't even include like the mandrel or the vacuum jacketing. I mean, or the leeway you need in the gyrotron to align it. It's an engineering challenge, and Chu Kuhn in the group, and Lynn and Alexander and Ronnie, that's all we think about, okay, lately. That's half of my intellectual bandwidth, is how do we fit a gyrotron into 15 millimeters, and how do we build this magnet? So it's a challenge, but we think that it's important for the field to not only have one option to get to high field commercial sources and high field stable NMR magnets. So we think that if we build the gyrotron for 15 millimeter diameter, that's going to enable us also to build NMR magnets further down the road.
Okay, and then the next question from Philip Grandetti. How does the rotor without the grooves or the fluid stay at the magic angle? I mean, this question has sparked, an interest, and so I see like 6 or 7 comments on it. So it has become a discussion now on the forum. So Xander, can you explain? Okay. Yeah.
So, a couple of it looks like it's been a little bit of question and answered. I think one answer to that is that you have this aperture. Let me go back to a slide. You have this aperture of, you have an aperture on the magic angle that sets the magic angle. Sorry, an aperture on the stator. So when the gas flows out of here, that's basically setting the angle. Where that gas flows, it's setting the angle without having the grooves on the rotor, because it sets, and then again, it's been kind of answered on the discussion. It's the friction. This is Tom Pop's argument. It's the friction along the rotor. And so he actually found these turbines that are only built on friction, if you look at this in the literature. So you can have you can impart quite a bit of power onto the turbine just by having the friction roll along the surface of the rotor.
Okay, and
is that answering it all the way, Kong? Feel free to look at this.
I don't know. Are you done with your answer, or you need to?
It is, but other people can keep on. Okay.
Okay. Okay. Let's go on to the next question from Matt Eddy. How do you calculate an estimation of the sensitivity needed to observe proteins in situ, for example, in cells directly? Does this consider the amount of protein present endogenously? Some proteins are expressed at only a few hundred copies per cell. What sensitivity enhancement is needed to observe such molecules?
Yeah, I mean, thanks, Matt. It's a really good question. I don't know what your GPCRs are. And as you have probably also, read and you already know, Matt, it's hard to actually determine that. And it's hard for me to find that in the literature. So I've spent, days, if not weeks, looking in the literature for protein kinase C, and the best I can come up with in the literature is about 0.1 micromolar. And so that compared to about what I look at in vitro, 10 millimolar, that's a factor of, 10 to the 4, we think. But I think it's going to be even worse than that, and I'd love to look at some GPCRs maybe together with you also. We want even more sensitivity than that. Okay, so I think the best answer to that question is whatever I think it's going to be, it's probably going to be way harder. And so that's why, for instance, I think that to really do good intact cell structural biology, that's why we actually want to detect on the electron spins themselves. So our strategy is to have an electron spin and spin it at high enough frequencies that the electron spin anisotropy itself is splitting apart into side bands. And then you're making a detection at 657, what is that, to the 5/2 better sensitivity, right? So we want to really increase the sensitivity of NMR by not just 10 to the 4, 10 to the 6. We're trying to imagine experiments that can get us to like 10 to the 12. And that would be, we hope, sufficient to look at the small amount of protein copy inside an intact cell. And again, let me just add, we really want to do endogenous levels of proteins. They're really nice work where you electroporate cells, very elegant. We actually want to take it to the next level and look at completely endogenous levels of proteins. We need even more sensitivity than we have today.
Okay, I just want to say, it's already 12:38. But I asked Xander before, since he's happy to stay on for more questions, because we have 50 more questions. So I will still keep on asking questions, and whoever wants to leave, I will thank you for attending this, and then after this I will send out the email and mailing list with all the recordings, and also I will also keep a copy. I can create a report of the Q&A, and then I will send to you. Okay. So next question from John. How do you get the spheres to 200 kilohertz? I don't know, like maybe like he asked like in terms of like what is the size of the diameter, and is it so light it will pop out and eject? How do you hold it?
I think my best answer to that question is we're going to do what everybody else has been doing, which is go small. So, you know, there are these 0.7 millimeter rotors, these great cylinders from Bruker that get to, you know, from what I hear, 111 kilohertz, 120 kilohertz. Of course, Agos Amundsen, probably the best in the world at making the cylinders. He gets to 160 kilohertz with, I think that's a 0.5 millimeter. So if the cylinders are doing 160 kilohertz with 0.5, we're always doing almost twice as good with the twice as high frequency with the spheres. So we think even a 0.5 millimeter sphere should be able to go north of 200 kilohertz. So going small, I think is my best answer to that.
So like someone also like follow up any question and asked like, I think in the slide he say like he saw like only 4 to 6 kilohertz MAS like were used. Is this like the spinning rate like the maximum at these cryogenic temperatures? I think like those 4 to 6 they were cylinders, right?
Yeah, so those are experiments. That's our limit at spinning at 4.5 Kelvin.
Yeah.
So we just can't spin higher than that. It's probably because we're like basically doing a helium fill on these cylinders, and there's a lot of helium blowing on it. And we've tried to push it hard. We can't get past 6 kilohertz when we're spinning at 5 Kelvin. Of course, we can get to like 12 kilohertz or 15 when we're spinning at about 100 Kelvin, similar to the Bruker technology. And that's for a 3.2 millimeter rotor. Of course, you know, they're the really nice experiments on using the Bruker instrumentation from Lesage and Elmsley and others that get above 40 kilohertz with DNP at about 100 Kelvin. And, you know, of course, you see better improvements with the homogeneous relaxation times on the nuclear spins. And that's really, you know, our inspiration for going to higher spinning frequencies. So I'm with you. We also want to go to higher spinning frequencies. Our strategy is to use the spheres.
Yeah. And then like the next question, do you have any idea why the proton T1 at 5 Kelvin or 6 Kelvin is so sharp? I think like what Xander showed was actually the T1 DNP, which is like 0.3 seconds. It's not like the actual proton T1 that you measure without microwave. Xander, can you like comment on this?
It's actually similar.
It's similar? If
so,
you just
as often you see, right? I mean, you know, tend to be more than I do about this, Kong, but from what we see when you have passively driven cross effect mechanisms, the DNP, the proton T1 rate is usually similar to the DNP rate for the cross effect. It's not always the case, but it's usually the case. And that's also what we see. So, and my understanding of that is that the relaxation sink on the proton spins is also still coming from the electron spin. So whatever couplings you have, which are connecting those protons to the electron spin, is either getting faster DNP or getting faster proton T1. What's the mechanism behind it? We don't know. I mean, the big difference on those tridal radicals is that there's some methyl groups that, I don't know, I mean, maybe they're still undergoing tunneling even at those temperatures. So maybe it's the methyl groups. We've started a collaboration with Matthias Ernst at the ETH to try to look at this with his dissolution DNP setup to try to, you know, model how that spin diffusion is kind of getting out so quickly. We think it could actually be useful for dissolution DNP. I wish I had an answer for you. I hope to publish a paper soon about it.
Yep. I'm just curious, like without the addition of the radicals, what is the T1 that you would expect at such a low temperature?
We've done that experiment. I think, Eddie, are you on? Do you remember that? I thought it was 40 seconds. Eddie or Nick did that experiment. It's around, I think it's 30 or 40 seconds.
All right. If Eddie or Nick, if you're around, you can type in the chat room, then everyone can see. Okay, I will go on with the next question. How the faster chirps will lead to further DNP enhancement? Which factor is mainly contributing to higher DNP enhancement?
We don't really know why the faster chirps are giving you better DNP right now on the chirps.
Okay.
I do know that if you have a higher gamma B1, then you'll be able to chirp back and forth and kind of drag that magnetization along better and have better control over the electron spins. So we don't know why the faster chirps are working better. Remember, this is solid effect, so it's not some like saturation experiment that I could give on the well, wait, It's tridal, but it is the solid effect. So yeah, okay. So maybe you're saturating better as you move faster over that tridal resonance. And then because you have better saturation, that's kind of driving the relaxation of cross effect better. I also encourage everybody to look at Song Yi Han's very nice, elegant work with the C equiball. I think they have some good understanding of the mechanisms of the spin physics that we didn't get into in our papers.
Yeah, exactly. I mean, this is also a very interesting question. Like Bob is also trying to do this here. I mean, if you can imagine, like all this, whatever drives the DNP mechanism is actually looking at the matching condition. And of course, you are doing magic angle spinning. So would the magic angle spinning not do the time modulation already? Why would you need an AWG or frequency chirp to do that? That was like kind of the question that people asked in the past before people want to do this chirp. So I think this is a new field. I think there's still a lot to be done. I think that we can probably outsmart just a simple CW irradiation, and doing anything like is always better than not doing anything. Yeah. Okay, next question: how tolerant are the smaller spheres to imperfect packing? Do the smaller spheres have stricter density requirement? How do you pack it?
Yeah, not yet is a short answer to that. So it's harder to pack the 2 millimeters just because they're small. Lauren, you can chime in on this too. She's the graduate that actually packed these samples. But what can I say? I mean, when we pack the 2 millimeter samples with KBR, which is the only thing we've done so far, they spin with about the same stability sample to sample as the 4 millimeters. So again, I think it's like the cylinders. You just need to make sure that the KBR is really particulate and you grind it up a lot. You need to pack it so it's really, jammed in there for the powder. I want to mention that we're not planning on doing powders for the rest of our life with the spheres. We want to look at proteins, we want to look at DNP samples, and then we're going to be centrifuging in samples, okay, like almost everybody else does in the world today when you look at small rotors. So if you're centrifuging in a membrane sample or a cell sample, then you're inherently going to have a lot better homogeneity and distribution of that sample inside the sphere, even for, let's say, a 0.2 millimeters rotor. We think we can pack 0.2 millimeter rotors because we're just going to centrifuge our samples into them.
Yeah, actually, I saw like Lauren, did you raise your hand just now? Do you want to talk? I just unmuted you. Do you want to talk?
I figured I'd help Professor Barnes out since he said something to me. You have to use the microscope. It's not horrible. Actually, the harder part is handling the caps. It's harder to handle the caps than to actually get the powder to pack. We've had multiple successes spinning KBR spheres at 57 kilohertz easy.
Okay, thank you, Lauren. Appreciate it.
All right, thank you. Next question from Dan. Typically, smaller rotors will eject without a cap or vacuum applied. Can you comment on this regarding the 2 millimeter spheres? On the 1.3, you have a cap, and then and then others, you have like Bernoulli valve system. Can you comment on this? How you can prevent it from ejecting from the stator for small rotors?
Yeah, those cylinders, man, they just keep on ejecting on you, right? It happens to us also. So with the spheres, we think that we have better fluid dynamics that are keeping the sphere within the rotor, within the stator. So of course, we've tried things that you might imagine where you have something like on top of the sphere to kind of keep it in place. It turns out that's not necessary if you design the stator in the right way to get the really good fluid flow to kind of suck the rotor back down with a Bernoulli action. Another thing I can tell you is that one of the first things that Tom Pop did in the group when he came is he got the spheres and he got the stator and he turned it around backwards. And it still spins. It still stays in there. So that really tells you that this gas flow, the fluid dynamics are really sucking those rotors in. So as Lauren told you, we've spun many samples at 2 millimeter and they don't eject from the stator. So we think that that's an advantage of the spheres as well.
Okay, next question from Keith. Many organic dyes act as efficient radical traps. Do you do anything special to prevent this in your link radical fluorophore?
Yeah, so thank you, Keith. Yeah, we've had the same huge concern. I mean, weeks in our lab, we talked about, what should the dipolar coupling be or how far away do we need to have that radical? And we picked to just have that big spacer on that TAT peptide. If you notice, it's like, 14 residues or so, 11 residues. So it's pretty far away. Our reasoning on that was that it's farther away than a fret pair. So we figured that that would be far enough away to not have that quenching mechanism. However, I'll also point you to Mark Bolus's 2019 Angavanta paper. He also uses fluorescent polarizing agents, and there he doesn't have a big linker at all. I mean, I don't think he has much of a targeting agent at all. It's only estimate 12 angstroms away, maybe the fluorophore from the radical, and he doesn't see that much quenching. The other thing I'll mention to that, Keith, is I don't know how much quenching there is, but optically, I mean, we got a lot of radical in there, right? I mean, we have like 5 millimolar, 10 millimolar radical within the cells, and that's a way higher concentration than you typically use when you're doing microscopy. So even if 99% of the fluorophore is quenched, which we don't think is happening, you'd still have enough to localize where they are.
Okay, I mean, like it's interesting. We have we still have a lot of questions, 11 questions, but I will maybe we'll keep the answer short. So like, how fast would a 2 millimeter cylindrical rotor would spin using helium? I think he meant like sphere. How fast would a 2 millimeter sphere spin using helium?
Well, I think he did mean cylinder. I mean, the sphere spins 57 kilohertz. It's a good question. You know, some of those. Again, Tom Pop showed me this really nice Andrew Lowe paper from like 45 years ago that showed a cylinder spinning with helium. I think it was like a 9 millimeter cylinder or something. And with helium, it's like ticking off to much higher spinning frequencies. So of course, helium works a lot better through cylinders as well. I think the reason you don't want to try that on a 2 millimeter is that it's going to explode. All right, so the mechanical advantage of the sphere allows you to leverage that better viscosity, the better effect of the speed of sound from the helium gas in order to access higher spinning frequencies without the rotor exploding.
Okay, and then the next question is like, so this person is getting confused. Are the spheres being spun at gas helium, pure gas helium, or a mixture of nitrogen and gas helium? When you say like you can spin the sphere twice faster, is it just using pure helium or mixture of nitrogen and helium?
Thank you. We can spin it at about twice the frequency of the cylinders when we're using helium. When we use air, it's still a little bit better than the cylinders, maybe 10 or 20% more. So a 9.5 millimeter rotors that are spherical, we can spin at 10.5 kilohertz with helium, about 6 kilohertz with air. The 4 millimeters, it's similar. We can do 15 or 20 kilohertz with air, about 30 with helium. The 2 millimeters, we can only get to spin with helium. So we don't have anything on air, but with helium, we can spin at 57 kilohertz.
Any prospect, great group asks like, any prospect of double rotation, i.e. a rotation around 2 angles, or even full rotation that's fast enough?
Yeah, so I don't think Tom Pop has the designs that he can share right now quite yet because they're not published. I can say that we're working on it. Is it promising? Yes, we sure think it is. We think that maybe the double angle rotation could really open up a lot of the periodic table to high resolution quadrupoles. We're looking into that. We think that the spheres would allow double angle rotation. And the other way that I like to think about that is spinning and processing about an angle very quickly. That's the only way I can really wrap around in my head. Yes, we think the spheres are possible. However, we're instead focusing on very stable spinning at really high spinning frequencies with more sensitivity. And this came from a conversation from Aaron Rossini. I mean, I asked him, you know, should I do double angle spinning? Maybe Claire Gray, you can tell me something else. I'd like to hear about it. You know, Aaron thinks if we just had enormously high sensitivity with DNP at really high fields, and one angle that was really super stable, you know, back to Dominique Mascot's question, very precise. And then you could do M2 MAS to get the resolution. He thinks that would be better. And I'm taking Aaron's suggestion on how to move ahead with it. Yet, we still do have some promising designs on the double angle spinning.
Okay, the next question, is the inside cavity of the spheres also spherical or is it cylindrical?
It is currently cylindrical, either bigger volume or smaller volume. We're also 3D printing some. Lauren has some designs 3D printed in ceramic that are more spherical.
Okay.
Let me add one thing. It can't be totally spherical because then you won't have that moment of inertia. So even when it's spherical, it's still kind of like a little bit thicker on the equator.
Okay. When can we expect to see DNP at 30 to 7 degrees Celsius? You have time frame for this? It's a difficult question.
I don't know. I mean, it's gonna happen in our lab. If we can do it first, it'll happen in 7 Tesla in our lab because we have a Gyrotron built already. We have the lenses built already. And we've had some snags with chilled water and et cetera at ATH. But, you know, I think we'll have our first good attempts. I don't know, Nick, what do you think? Like October, you know, December. I hope that Bob or Song Yi can beat me to it so I can just follow what they did, or others. That'd be great. But, you know, I think we'll have some good attempts within the year. I think we got some good shots in the next 2 years. And if it takes 25 years, I'll damn well do it.
Yeah. I mean, Bob, you can also comment if you want, you know, you're a panelist, you know, if you want to add on to this, it doesn't have to be just Xander and I, right?
Well, there have been many DNP experiments at room temperature. The very first one we published with the Gyrotron was BDPA and polystyrene. Of course, it's polystyrene sample, so it's a low dielectric constant sample. But there are DNP experiments at higher temperatures. You get much smaller enhancements. You get 10 rather than 400. But it's possible. You know, we just need to we need to get the instrumentation together to work on it.
All right. Next question.
How much? Can I add a little bit more to that?
Sure, sure.
I mean, yeah, there have been amazing papers from Bob's lab. I mean, you and Kong, the panelists are getting factors of 200 or 300, you know, at room temperature static. I think the big challenge, in my opinion, is doing this with match angle spinning, right? Because in Kong, all of your experiments, many others, you know, Bengenbach, many others, you always use these resonators to get the quality factor and the gamma B1 on the electron spins. So if you could have those resonance structures, you know, maybe there's some other ways that you guys are thinking of at MIT. I'd love to copy them once you publish them. If there's some ways to get those cavity structures, then that would be great. The difficulty is doing this with match angle spinning.
Yeah, exactly. You're right about this. And then, like, you know, like with the match angle spinning data, you have, like Bob always say, oh, you have a Q of 1. You know, if you can have a Q of 100 or 1,000, it's either you improve the Q factor or you improve the microwave power. Of course, you don't want to blast like megawatt or keep like megawatt of microwave power into your sample. So like it's one way or the other. Yep. Next question. How much helium would you need for 100 Tesla magnet?
Well, we're making small magnets. So I think it's going to be a lot less than Bruker's using for 28 Tesla magnets. So our strategy is to make these tiny magnets, and they're going to be a lot smaller, and I think it'll be a lot less helium than you're currently using for these high field, extremely large magnets. So we're optimistic that we can make these things efficiently, but you don't have to believe me on it. That's okay. I'm going to try to do it anyways. My group is working really hard on this. I don't know if it's going to take us a year or 10 years, but we're really dedicated to trying to figure out how to do that.
Yeah. And how much heating occurs in the samples at higher spinning rates? For a biological sample, what is the faster MAS likely possible that will give a sample temperature of 37 degrees C?
So, you know, we measured with the 9.5 millimeter sphere spinning at 10 kilohertz. We got an IR thermometer and put it on the outside of the sphere. And for those spheres, we couldn't detect a temperature increase. Of course, you'll see in the literature, this has been discussed a lot by many pioneers, many really good groups. You get a lot of heating on the bearings, typically, where you get a lot of friction. And so what's typical that people do is that you use colder bearing first, and then the friction kind of heats it back up to room temperature is what typically is done in the stators. So we think that something like that can be done on the spheres as well. So for instance, you could, you know, slowly ramp up the spinning frequency, slowly cool down the bearing so that you kind of maintain this sample temperature that would let you do something physiological.
And then like the next one is just a comment. Like someone from K group like say that they did like lithium overhouse DNP at room temperature. Actually, I was actually there like in ENC Baltimore and I listened to this talk. And then yeah, I mean it was interesting. And then I think it was Michael Hope, who is a postdoc now at Linden Ansley Group, and they use 600 MAS DNP to use to do the overhouse DNP. I mean that's the first one that was shown the DNP effect. And also like of course Aaron Rossini, like they also did something with a room temperature DNP. like on the gamma irradiated quartz, but it was a special case. So these are the comments that I got. All right. Then next from Ivan.
Something about that, because I mean, look, there have been a lot of great experiments. So in no way am I saying that there have been a really nice experiments that have been done at room temperature with DNP. I mean, Jake Schaefer also did some really nice experiments with BDPA earlier in the 90s. I think, and Aaron's experiments are really elegant with the super long relaxation times on the gamma irradiated quartz. Look, the challenge here is to do pulse DNP on biologically relevant systems on like a membrane. So what we want to do is pulse DNP on a radical with a really, that we can get into our systems of interest for material science or for biology. And for that, the electron T1s are always going to be short. Okay, we want to look at a system that does have short electron relaxation time, and for that we need a pulse and strong hyperfine interactions to transfer that polarization.
Okay, we have 7 more questions. So Ivan from Brooker asks, at fast MAS and higher electron nuclei B1 fields, what would you expect to happen to the efficiency of direct DNP mechanisms to low gamma nuclei?
I mean, kind of like Con said, we'd like to get into this regime where it's just like the protons to the carbons, Ivan. So if you're spinning at 500 kilohertz, and that's even faster than the hyperfine coupling, but you can affect the electron spins with pulses, then if you do CP, we'd expect a really good transfer. So we'd like to do the same kind of methodology using solid-state NMR, except just using the electron spins. I need to add, of course, this has just been driven by Bob for the last 30 years. It's been his dream to really have pulse control over the electron spins. But we think the key is really a really good gamma B1, Ivan, on those electron spins. The technology for that doesn't exist, so we're going to build it ourselves.
Yeah, exactly. I mean, with a low gamma nuclei, the main difficulty is about like polarizing the bulk, because like the spin diffusion is not very efficient. I mean, there are a lot of ways, some ways you can do a CP or through the proton or direct DNP mechanism, but that's like sometimes not very efficient. So, but these are, I think this is an open question that a lot of things can be done for DNP application on materials. Okay. What duty cycles limit do you expect for your pulse and high power gyrotrons or amplifiers? Would this pose a problem for pulse DNP?
That's a really good question. Our duty cycle on our gyrotrons is 100%. And we want to maintain that. We think it's really important to have 100% duty cycle on the gyrotrons. So everything that we do is 100% duty cycle on our gyrotrons. This is one of the reasons why we think that gyrotrons are really the way to go. So gyrotrons can give you a lot of microwave power at 500 gigahertz, 800 gigahertz, should be, even higher terahertz. And they can also give you really high duty cycles of microwave power. So we really think that gyrotron oscillators, and then either doing chirps or using these splices and semiconductor switches, again, following UCSB with Mark Sherwin et al. We really think that that's the best strategy to do pulse DNP in the future. And then, if you don't need the microwaves, you can always just turn them off by switching the voltage on the gyrotron quickly, or you can just activate a switch to shunt the microwaves. But we'd much rather have 100% duty cycle available. You can do that with the gyro amplifiers also if you designed the vacuum and the cooling and the collectors. So both of that technology is available. We just think that the gyrotrons are going to scale better up to 800 gigahertz, 1100 gigahertz in the future.
Yeah, maybe I should say, like, from the aspect of like gyrotron instrumentation. So like, so it can have either like 100% duty cycle with 100 watt, or you 100% 1 watt, or you have 1% 100 watt. Is the same. It only depends on your cooling capacity. So you can find a compromise, because like in principle it should not affect like the gyrotron operation, but of course it's only determined by your cooling. Okay, what is the temperature effect of your microwave decoupling on the sample, especially in your case when you use very short T1s?
Yeah. Okay. So really good question. Of course, we've been asking and answering it a lot in the group over the years. The short answer to that is that we can barely detect the temperature increase with the microwaves on in the sample. So Nick and Eddie, again, if you guys are on, you can comment on it. We've done the experiment, which is one of the best experiments, which is to measure the T1 using Kent Thurber's and Tico's strategy of looking at the KBR relaxation time. So we look at the KBR relaxation time with and without the microwaves. And when you're blasting the helium or the nitrogen on the center of the rotor, we barely see a temperature increase. So I think it was undetectable, or maybe half a Kelvin or a Kelvin. So again, if you have so much cooling power on the sample, it's actually not a lot of sample heating. And part of that is also driven by the dielectric constant of the sample, right? So, you know, why does it take you so long to microwave that frozen lasagna in your microwave? That's because the dielectric and the loss tangent is so low of ice. If you do this at room temperature, that's going to be a lot bigger concern because the microwaves will absorb a lot more. But in short, we barely see a temperature rise in our sample. We've done that with NMR thermometry.
Yep. I think like we already like past the time. It's already 1 o'clock something. I will just take like one last question because I skimmed through the question. Like a lot of these like things you can find like in his papers. So I will just ask the last question and then like just call it off. And is that okay?
Yeah, sounds good.
Okay. So like I just pick one question. What about the critical field strength of the superconducting tapes towards 50 or 100 Tesla?
So there's a nice paper or talk by Yuki Iwasa, and he thinks that you can have critical, you can have magnetic fields up to 100 Tesla, you know, optimistically. So you're going to be bordering that limit of the critical current. But this Rebco tape, it's already really good, and there have been some preliminary results in the literature where you can get better pinning on the Rebco tape, which should be able to get even higher critical currents. So as it is today, we think that we almost should be able to hit those really high magnetic fields, but in 5 years, that technology is only going to get better and better.
Okay, I mean, like this was going on, and then like, I didn't notice this, and then like, I should have launched a poll. So instead of having 250 participants that we originally had, we only have 100 participants left. And I have some, you know, a poll you can see here. Feel free to vote on it, and so that I can collect feedback, and so I will know how to improve it for the future series of the seminar. And you can take your time to do this. And I mean, Bob, you want to say something? Thank you. Whatever, Bob, are you still there?
Yeah, I'm still here. And yes, thanks to everybody for participating. I think we expected maybe 50 or 60 people. Today we had, because of Xander's great talk and so forth, we had over 250, up to about 255 or so. So this was, we'll declare this a success, and we look forward to the next presentation next Wednesday. I think the time may shift a little bit. From say 11:30 to 10 o'clock or 10:30, something like that. We'll let you know so that people in Israel can not interrupt their dinner and still watch the presentations. Okay, so thanks again, Xander. That was a really fantastic talk. You got a lot of feedback and good questions for it, and so you really launched this effort on a great trajectory, we hope.
Thank you.
Okay.
All right, thank you. Yep. So like as Bob mentioned earlier, like we would like to encourage like more students and postdocs like to submit an abstract. Actually, I created a website like just yesterday night, and then like all this like information will be announced through the mailing list. So so we can want to get some feedbacks. We will probably try to squeeze maybe like put 2 or 3 talks per session and then make it 1.5 hour long or something like that. So we will decide after looking at the post and feedback. So thank you, Xander, very much for your kind contribution. And it's an amazing talk, and I really enjoyed it. And I don't know, looking forward to see you in Europe or in Zurich or whatever. Thank you very much, and see you.
Thank you for putting all this on, Kong. Thank you. Bye.
Okay, bye. Bye. Stop video. Stop recording.